Hello, my lovely anatomists and physiologists. Michele here, back with chapter 12. We left off with our neuron, which is our functional cell type, the nervous system cell that's doing communication. And we're here with what are called glial cells. Sometimes they're called neuroglia or neuroglial cells. These are going to be more like your supporting cell. And we have 6 types. We have 4 that are found in the central nervous system, and then we have 2 that are found in the peripheral nervous system. The central nervous system, the CNS, remember, it means your brain and your spinal cord. So let's look at these together. Let's start with your astrocyte. Your astrocyte is going to be, really just important as a support cell. It's going to maintain the concentration of chemicals in the extracellular space. Is going to help remove excess signaling. Molecules. They're going to help react to tissue damage. And then they're going to be important in forming what's called blood-brain barrier, which is abbreviated as BBB. And we'll talk more about that in just a few minutes. So really the astrocyte is helping to regulate, what chemicals have access to the neuron, or you can reverse that and say what chemicals the neuron has access to. And also important in helping to do some repair. The next type of cell is called the Allego Dendrocyte. The Allego Dendrocyte is going to form myelination. And a key feature here is that it can myelinate multiple axons. Myelination, remember, is that kind of insulating wrapper on the axon that helps to speed up the propagation. So that's going to be really significant. We also have in the central nervous system what's called the microglia. And the microglia cell is basically the phagocyte that's resident to the central nervous system, so the brain and the spinal cord area. So it's doing all the actions of phagocytosis. So phagocytes here are going to be important in cleaning up debris if there's any kind of damage, and also engulfing pathogens. So this is going to be an important defense and cleanup cell. Let me put that in parentheses, defense and then cleanup. And then the last one for us to look at is called the epidermal cell. And the epidermal cell is going to make what's called cerebrospinal fluid. You can abbreviate cerebrospinal fluid as CSF. It's also going to circulate CSF. So this is going to help it wash over the cells of the central nervous system. When we talk about it making cerebrospinal fluid, let's make sure that we know that it's filtering and absorbing components of the blood. to make CSF. And then we'll see that the epidermal cell is going to line what are called your brain ventricles. Line your brain ventricles. Okay, so the astrocyte, Allego Dendrocyte, microglia, and epidermal cell are the glial cells that are found in the central nervous system specifically. When we talk about the peripheral nervous system, we have just two types of cells here. We have what's called the satellite cell. And the satellite cell is going to be found in sensory and autonomic ganglia. We've defined the term ganglia as the collection of cell bodies in the peripheral nervous system. So the satellite cell is kind of doing for the peripheral nervous system neuron what the astrocyte does for the central nervous system neuron. They're going to surround cell bodies. They're going to be like the astrocyte, but no blood-brain barrier because we're not in the brain anymore, right? We're in the peripheral nervous system. And then the other type of cell here is the Schwann cell, and that is spelled with a capital S. This is after an individual. The Schwann cell is going to form the myelination in the peripheral nervous system. And all of the axons in the peripheral nervous system are myelinated. And here's the key difference between the Schwann cell and the Allego Dendrocyte. The Schwann cell can only myelinate one axon. Whereas remember, the Allego Dendrocyte could do two. So we have the astrocyte in the central nervous system doing kind of a similar job as the satellite in the peripheral nervous system. And then we have the Allego Dendrocyte in the central nervous system acting similar to the Schwann cell in the peripheral nervous system. Let's dig just a little bit deeper on our blood-brain barrier and our myelination. So remember, the blood-brain barrier is formed by the astrocyte. And it's going to restrict what can cross from the bloodstream into the central nervous system, into the CNS, so the brain. So remember, we can abbreviate blood-brain barrier as the BBB. So when we talk about what can cross, so I'm going to do like, yes, can cross, no, can't cross. Things that can cross would be glucose. And this is actually the primary energy source for your neurons. Primary energy source for your neurons. We can get amino acids, we can get water, we can get gases, so that's CO2 and oxygen, and we can get ions. All of this can cross. White blood cells can't cross. And so this is the reason for that microglia, the resident phagocyte. So we're not able to have our defensive white blood cells actually crossing into the brain and the spinal cord area. And then lots of drugs don't. And here, drugs, we're talking about like pharmaceuticals. Hello, my lovely anatomist and physiologist. And so when we're talking about drugs here, we're talking about pharmaceuticals. So this is a barrier that pharmaceutical companies and physicians and pharmacists have to deal with. How can we form drugs that, you know, if we need a drug to get to the brain, how can we cross that blood-brain barrier? The other topic here to talk about a little bit more is the myelination. And myelination is going to give us the white matter to the nervous system, and that's going to be true for both the central nervous system and the peripheral nervous system. And this is because it is a lipid-rich sheath. Lipid-rich sheath. And so it's given us this lighter color appearance to this nervous system tissue that we can see, in dissection or in surgery as having this whitish colored appearance. Remember, it's being produced by the Allego Dendrocyte and the Schwann Cell. It's increasing the rate of action, potential, propagation. And what we'll see is that these cells will actually wrap themselves around the axon, and it's actually the cell membranes wrapping around the axon. It's described sort of like a flaky pastry. So layer, 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 like that wrapped around. And it's maybe 1 to 2 millimeters in diameter around. And so as we get into our conversation of how action potentials propagate, then we'll have a better understanding of how myelination is speeding up that transmission. You do have in your text a description of some disorders, diseases that occur that actually break down this myelination and how it can affect an individual. So that can really help emphasize when it goes wrong. That helps us see what it's doing for us when it's working right. So that can be interesting to read. Okay, as always, take care of yourselves and each other's, and stay tuned for the next video.